Hi there. Okay, so this is the third little video, so we've already talked about vacuoles and plastids, and now we're going to talk about the outer surface of the plant cell. So it's a layer, it's called the cell wall, outside of the plasma membrane. We're also going to talk about the cell-to-cell connections called plasmodesmata. Okay, those are important as well. Now, the cell wall is made up of a whole bunch of different components. It's not just this inert bunch of cellulose to the outside of the cell, but it does a whole bunch of things, and it has a whole bunch of components. So many functions. It supports the cell. It prevents it from blowing up, which is kind of important, right? Kind of constrains expansion. It will determine the shape and size, act as a barrier while still allowing the transmission of signals and hormones and water through the plasma membrane. And it's kind of the first responder to external stimuli. And in some cases, as we'll see, it's involved in storage. So it must allow all these things to happen, and it must allow materials to go in and out of the cell. Now, the cell wall is laid down often in two stages. So we have what's called the primary, which is laid down first, and this generally happens while the cell is still growing or expanding. And then we have a secondary wall. Now, not all cell types have a secondary wall, but in some cases, after growth is complete, other layers of cells are laid down, and this component is actually often lignified, has lignin in it, and we'll talk about that. So the first component that we're going to talk about is the middle lamella. It's actually the layer that glues the two cells together. So here you can see we have cell number 1, and we have cell number 2, and then they're kind of glued together by the middle lamella, which is made up of a chemical called pectin, which is a very complex polysaccharide. It is hydrophilic, so it absorbs water. Now, as, say, fruit ripens, it gets softer, basically these pectins are broken down enzymatically, and the tissues kind of fall apart. So, think about eating a banana when it's not ripe. It's kind of hard, right? But then as it gets ripened, it actually softens up. Pectin is one of these things we call a soluble dietary fiber. So it actually dissolves in water, but it's still not digestible, and it helps like little tiny scrub brushes cleaning out our little intestines. Now, as I just mentioned, pectins are abundant in fruit, and they're actually extracted from many fruit parts and used commercially to help in the gelling when you're making jams and jellies. So kind of nice little practical application of pectins. Now, the primary cell wall, where we'll find externally to the plasma membrane and internally to the middle lamella, is made up of a bunch of different components, including cellulose. So that's the major one that we're all really familiar with. So we're going to talk about that one first. And it's made up of, it's actually a polymer of glucose molecules, and they're in a linear array. Now, when we think of cellulose, they're not just these polymers, but they're actually a collection of the polymers all together to make one cellulose microfibril. And now, cellulose is the most abundant organic molecule in the world. So since plants generate lots of this cellulose, plants are generally a really, really great carbon sink. Okay, so what I'd like to do actually is assign you a reading, pages 60 and 61 in the 8th edition of the textbook, and page 56 to 57 in the 7th edition. And I'd like you to read those pages and think about how cellulose is actually deposited into the cell wall. Okay, so I guarantee there will be some kind of question on the midterm about that. Okay, so what do people use cellulose for? Well, of course, we know we use cellulose for many things, including paper, but clothing, like this is a cotton plant, and cotton actually is fibers, cells from the developed seeds. And this is a cotton boll, so that's the fruit, and then these are all the hairs on the fruit. Very interesting. But things like cellophane, you might not even think about it, but cellophane gets its name from being derived from cellulose. And this made this kind of a viscous substance, which is made from wood and cotton and hemp, and makes this plastic-like stuff. Rayon is something similar. It makes this viscous-like stuff. They press it through the spinneret, kind of like a shower head. They kind of liquefy it, put it through the shower head into a bath where it solidifies into these fibers. So rayon is actually a semi-synthetic type of a fiber. We also have dietary fiber as well. Cellulose is a non-soluble one. Also in food additives, if you read the ingredients on some of your food, you may see methyl cellulose, which acts as a food thickener. It also acts as a laxative. So, two things for the price of one. Also, there's lots of research right now on biofuels and taking a look at good sources of cellulose, which could be broken down into sugar, of course, which then could be fermented and fermented into alcohol, which can be used as a biofuel, bioethanol. Okay, so another component of these primary cell walls is the hemicelluloses. So this is the little red lines here that are acting as crosslinks. So they're actually help to keep those cellulose microfibrils in place. There are different kinds of sugars. It's sort of more of a heterogeneous type of molecule. And you can also see this pectin is in the primary cell wall as well. But you'll also notice there's lots of space in here. So there's lots of water can get into the cell walls. As I mentioned, sometimes it's storage. So sometimes these cell walls store water. And if you think about it, water could also be passing through the primary wall. So it doesn't act as a barrier to the movement of water. So the pectins and the hemicelluloses, they're actually deposited a little bit differently than the cellulose. The cellulose, of course, has enzymes. There's enzymes embedded in the plasma membrane, the cellulose synthase enzymes, and the microfibrils are generated by these enzymes in the cell wall. The pectin and the hemicelluloses are synthesized in the Golgi and secreted by secretory vesicles, fusing with the plasma membrane and then into the cell wall. So a little bit different. We also, here's a slide showing the cellulose microfibrils with these cross-links of the hemicelluloses. We also have things like proteins. So different kinds of proteins. There's enzymes. If you think about if a cell is growing, you're going to need enzymes to cut some of those cross-links to allow for slippage so that your cell wall can loosen, so the cell can grow. We also have structural proteins as well, glycoproteins in the primary wall. And as I mentioned before, water. And if you think about it, the cell walls have lots of pectins, will have lots of water in them as well. I want you to really think about this idea of water movement because it's actually important when you think about the movement of water through tissue. We'll talk about that particularly when we talk about roots. Water can move in two main ways. One, through the cell wall. There's sort of a continuum of cell wall. And so water can move that way, and that's called apoplastic water movement. And then we have symplastic, because we have also movement from protoplast to protoplast through these cell-to-cell junctions I mentioned earlier called plasmodesmata. So you can see here's some plasmodesmata right here in a primary pit field. So primary pit field is just sort of a thinning of the primary wall, and then there's these plasmodesmata running through. So larger molecules can run through, those that can't, say, go through the cell wall and things like that. So kind of important. And so here's a picture here showing you, this is actually really, really thick primary wall. You might think of primary walls being thin, but sometimes you have nice thick ones. And here you can see the middle lamella gluing those cells together, but you can also see these plasmodesmata running through this primary wall. Now this slide was stained with toluidine blue, a stain that you're going to use in a lab, and when you see the stain reacting and turning purple, then that indicates that your cell wall is mostly cellulosic. If it was kind of a baby blue color, that would indicate that it had lignin in it, which could mean a secondary wall, which I'll talk about in a minute. So just thought I'd show you that for now. So here's a primary pit field, here's these plasmodesmata in between those two cells we looked at earlier. And just a few more slides just to show you various thickenings of the primary wall here, but then you have these areas that have the plasmodesmata. Now, the plasmodesmata actually are lined with endoplasmic reticulum, which is kind of neat, that runs from cell to cell. So they're actually really connected. Larger, as I said, molecules can pass through. In fact, viruses can pass through. That's how plants get infected with viruses. It spreads through the plasmodesmata. Okay, so before we get on to talking about the secondary wall, I just want to mention that a cell that has all the components that we have just talked about, it can still grow. And remember that cell growth, there's two things. We talked a little bit about the cell wall needs to be loosened, so there's enzymes that are going to help snip some of those hemicellulose bonds. But we also have the vacuole expansion that helps to drive that growth. Okay, so we're going to talk a little bit more about that later on. Now what I'd like to talk about is the secondary cell wall. So some cells, not all cell types though, some cells, we'll talk about very special ones, after the cells have finished growing, will deposit a secondary wall. Okay, so after growth is complete, another wall is laid down inside of the primary cell wall, called the secondary cell wall. And it's really rich in cellulose and hemicellulose, but not much in the way of pectins or proteins. Another thing that's added is lignin. And lignin fills in the spaces within the cell wall. So in the primary wall you have all that space. But in the case of the secondary wall, all those spaces are filled in with lignin. And that's really what makes wood so strong. And also it helps to waterproof that part of the wall. So water can't actually flow through. And that's going to be really important when we talk about some of the functions of these cells that have secondary wall. Now, one thing about lignin is that once a plant deposits lignin, it actually can't take it back. It can't degrade it. The only thing that can degrade it is fungi and maybe some bacteria. So the cell actually no longer can grow or divide. And sometimes the cell is performing a function because of that thick cell wall. And so actually at functional maturity, those cells end up being dead. So, yeah. So once the cell wall gets lignified, there's no going back for those cells. Now the cell wall, as I mentioned here, is laid down in layers, and you can see there's a 3-layer cell wall, secondary wall. I don't really care about the 3 layers, just so that you realize that the wall is laid down. Here's the plasma membrane would be here, is laid down between the plasma membrane and the primary wall. So this is the primary wall. And that's kind of an important thing to know where these walls are relative to each other. Okay, so here's the example here, and I know you're going to say, oh, it's purple. Well, this actually wasn't stained with toluidine blue, so, but these are secondary lignified cell walls. Okay? Now, lignin itself is a polymer polyphenol. So here there's sort of 3 different kinds: coniferyl alcohol, coumaryl alcohol, and synapyl alcohol. And I'm never going to ask you the structure of them. Just take a look at the big mess they make when they're all linked up together. Okay, and really tough, as I mentioned, to break those bonds. Something kind of interesting, coniferyl alcohol is found in conifers. That's not a big shock. And so you can sometimes tell what kind of wood you're looking at based on the proportion of these different alcohols. So for example, conifer lignin is made up of coniferyl and coumaryl only. Monocot is an even mix of all 3, and then coniferyl and synapyl alcohol are mostly common in eudicots. So that's what this is. So it's probably eudicot lignin. Now, one other thing I think is really important is we have our secondary wall laid down here. But, as that wall is being laid down, the cell has to be able to communicate and pass things between other cells. So nutrients and water have to pass between cells. And so we have to maintain these avenues. And these are called pits. Did you notice the pits are actually not holes? Okay, they're not holes. They are, there's thin areas. They're areas where you don't have secondary wall, but those primary walls are still there, and these are usually over areas where the plasmodesmata are. Okay, so that's kind of an important thing. Okay, and the area here, of course, remember your primary wall is going to allow water to go from one cell to the other. Okay, but water's not going to pass here, remember, because that's a secondary wall that's got lignin in it. Okay, so I think we'll end there. I just wanted to show you this very beautiful picture. This is the town I grew up in, and this is the pulp and paper mill right here. And so remember, paper is basically made up of cellulose. Most of the lignin has been removed, and to remove it takes very toxic and stinky chemicals like sulfuric acid. So my town, even though it looks really beautiful, sometimes is a little bit on the stinky side. Okay, thank you for your attention, and we'll see you in class. Bye.